With that, John, we look forward to your seminar.
Okay, thank you, Jerry, for the introduction. Again, I'm John Fulton at Pacific Northwest National Laboratory here in the state of Washington in the beautiful Pacific Northwest. It's a great pleasure to be able to tell you our recent story about ion ordering in solution for this series. I have a long interest in using X-rays to measure the hydration of ions and look at the interaction of the cation with the anion. And today, all the focus of the talk is more on the long-range ordering. The title is Ion Correlation X-ray Spectroscopy, and what I mean by that is using various X-ray methods to measure the ion-ion pair correlations in solution. This comes traditionally from X-ray scattering, but of course, XAFS is ideally suited for this, and you can get indirect information about the pair correlation functions from things like XANES or X-ray emission. So we have a little technical problem here. It was working quite smoothly here, but we're having trouble advancing. So we'll try to stop sharing and reshare.
We have a good view.
That's right. Okay. So in the tradition of this series, I've three different sections with questions between. I'll start out with sort of a overview of the various definitions of ion-ion structures, move on to some discussion of the context and motivation for this work. And then finally, I'll take a little bit of a look back on how we used X-ray absorption spectroscopy to measure the short-range structure of ions in solution. The bulk of the talk will be on using SAXS, especially analyzing this pre-peak here to give us information on the periodic ordering to longer distances. And in the final section, I'll talk about looking for the Kirkwood transition. This is something proposed by theory that it's a certain salt concentration around very roughly about 0.5 molal. And molal is the number of moles per 1,000 grams of a solvent. It's a convenient measure. At a certain concentration, we go to a system that shows periodic ordering to long distances between 1 and 5 nanometers. So to begin with, this is a team effort, of course. There's myself and Elisa Biasen who are doing the X-ray spectroscopy on these systems. We have a theory group here at PNNL who has like interests in looking at aqueous systems, and that includes Sean Kathman, Greg Schenter, and Chris Mundy. I'd also like to highlight the contributions of the postdoc in the theory group, Bahati Dinpaju, here, who has done a lot of the heavy lifting on the theory part of this analysis. We've had contributions from Abdullah, other postdocs, Abdullah, Nadia, and other staff contributions from Emily and Sebastian here at PNNL. The work is funded by DOE Office of Basic Energy Science. This is primarily a lot of SAXS studies, and we've done measurements at a number of different beam lines. In fact, we've visited 4 different beam lines at APS, And currently, Elisa Biasen is making measurements at SSRL at beamline 4.2 with Thomas Weiss. And if you're at moderate concentrations, 0.5 up to very high concentrations, this is very relatively easy to get a measure of the SAXS spectra, these pre-peaks. But when you go down to dilute solutions from 10 millimolar up to 500 millimolar, the scattering is extremely weak. And in comparison to a standard SAXS experiment where you're looking at colloidal materials, nanophase materials, the scattered intensity for these systems is at least 2 orders of magnitude weaker than what is normally experienced in SAC. So we've been very careful to run multiple replicates of the measurements at numerous beam lines and look at a series of different salts with a range of concentrations to make sure that we understand the signal response. And we can classify the interaction of a cation anion over a broad length scales. Commonly, we talk about the contact ion pair, which is the direct interaction of the cation with the anion. Another very familiar species in solution is a solvent shared ion pair, where the distance between the cation and the anion is separated by a couple of water molecules. And then finally, the interest for this work is to look at the longer range correlation of these ions in solution. So the method of choice, of course, I have a strong personal bias. I've been doing XAFS for many years. And for contact ion pair structure, the method of choice is XAFS. You get a very clear measure of the bond distances, the coordination numbers, and the Debye-Waller factor. If you're looking at solvent shared ion pairs, this is a longer distance, probably out of the reach of XAFS. So here we have method of wide-angle X-ray scattering, also known as X-ray diffraction, and more recently termed PDF method. You can also get information on this longer range structure from Zane's calculations. And the method for these long range would be small angle X-ray scattering. This is, of course, an X-ray absorption spectroscopy series. And I would propose that, and I'm firmly in the camp of using X-ray absorption spectroscopy, but I would propose that there's things that we can learn from scattering. I know there's in my experience, there's two different distinct groups. There's the absorption group and there's the scattering group, and they don't communicate that well. So I would suggest that in order to make progress in this long-range ordering, that we need to not only understand the speciation at short range in order to understand the response of the system at long range. So we can make use of this both types of information. So the importance of these systems is that in almost any aqueous system on the face of the Earth, ion correlations are important. In the liquid phase, it's critical to understanding the mechanisms of biochemistry, membrane transport, cell functions. The recent interest in batteries for not only transportation and grid storage, these are all based on the properties of electrolyte ordering in solution. In hydrothermal, in concentrating of metals in the earth crust, the concentrating of rare earths are all based on hydrothermal transport of these materials. In industrial chemistry, we have importance in the area of hazardous waste treatment and the power plant water chemistry. And then finally, there's instances in atmospheric chemistry, aerosol catalysis, marine aerosols, where these ionic species play a major role. To give a little bit of history here, this is more or less the state of the art as far as XAFS versus wide-angle X-ray scattering. This is some measurements of 5 molar cobalt chloride in water. The green spectra here at the top are from wide angle X-ray scattering. We have XAFS measurements at the cobalt edge and at the chlorine edge. And what we find is excellent agreement between these two. And in fact, the cobalt water distance here for X-ray scattering versus XFs is within a few thousandths of an angstrom. We have excellent agreement with the Debye-Waller factor. And likewise for the chloride structure, the chloride water distance is within a little less than 100th of an angstrom in agreement. The thing that's probably not as well understood, at least for these aqueous systems, is that the spatial resolution for XFs is typically much higher than what you can get for from wide angle X-ray scattering. So the measurement of these oscillations out into Q or K space in XFs is typically to a much higher momentum parameter than for in X-ray scattering. So there's an advantage with XFs, better spatial resolution. The other problem with wide angle X-ray scattering is that you're looking at contribution of all the different partials in the radial distribution function. And that's a problem because the water-water structure often interferes with your interpretation of the position and the assignment of these peaks. The beauty of XAFS is that you can go in and look at a particular absorption edge and probe just the pair distribution function of that particular ion in solution. So in this case, I think, and again, I have a certain bias here that XAFS is the hands down winner for this short range structure. Giving you a little bit more background information here. This is a study that we did some years ago where we used chloride XAFS as a so-called a universal probe. So we looked at the complete series of alkali and alkaline earth cations in solution and looked at their chloride salts. We did chloride XAFS then to try to detect the presence of the contact ion pairs for this series. We also backed that up with wide angle X-ray scattering to evaluate the existence of longer range structure. So these are the radial structure plots from XAFS at the chlorine edge. And this main peak here at about 2 angstroms is due to the 6.4 waters that are hydrating the chloride. And what we see, these are saturated solutions of salts. This is the alkaline earth cations here at saturation point. And what we see for all of these divalent cations that we do not disrupt the water shell around chloride. So we believe that if the contact ion pair will displace the waters in the first shell and disrupt this hydration shell. In contrast, for the alkali cations, the monovalent cations, for lithium, we see the full complement of 6.4 waters, but as we go up in the series up to cesium, we see increasing loss of water in the first shell. This is indication that for these monovalent cations, we form contact ion pairs. And in fact, these are the positions, these arrows I've added here show the positions of the backscattering from the nearby cation. And there's clear signals in detailed analysis that would correspond to the cation in that first shell. And in fact, we've also paired this with wide angle X-ray scattering that gives us the longer range view here. So these are the XAFS experiments that I showed, but for the same series, we notice that for divalent cations, we see evidence of the solvent shared ion pair, whereas for the monovalent cations, there's very little existence for the solvent shared ion pair. And this can be summarized in this simple plot here. If you have a high charged cation with a small radius, you have a very high local electrostatic potential. And in this case, water is the favored ligand that competes for the anion in the first shell. And if you're above a certain charged ion radius, we have predominantly a solvent shared ion pair. If you're below this, you'll have a contact ion pair. Of course, this relationship is for chloride as a counterion. If you use a different counterion, you probably move the position of this line as it affects the quality of the anion as a ligand for the first shell. So I think that sort of wraps up the introduction part. So if I've left anything out, if there's any questions, I'd be glad to answer those.
No questions quite as yet. So I guess one very simple question, and this is just proving your point about the chasm between diffraction people and XAFS people. What you're calling wide-angle X-ray scattering, how similar or different is that from what people would call X-ray PDF, that is pair distribution function? I see that you're doing that analysis, but are you up at 100 kilovolt photon energy, or is there any difference?
So I consider I think that the old literature would talk about X-ray diffraction, X-ray scattering, and then so we're looking at Q values from probably 2 up to 15 or 20. I consider that the wide angle X-ray scattering region in contrast to small angle wide angle. So and also, I consider the use of PDF as sort of a recent terminology that perhaps gives a better visualization of what you're measuring, but the basic methods I feel are the same as the old historical use of X-ray diffraction or wide-angle X-ray scattering. So it's my personal interpretation.
All right, that's fair. And I'll paraphrase a question from the chat. Was there any possibility in these systems of doing anomalous wax to try and separate out the different partial pair distribution functions?
Yeah, so my colleague Elisa Biasen has been working on doing anomalous scattering, both primarily anomalous SAXS, so looking at things like rubidium and strontium and getting very specific information on the various individual partials. So that's an ongoing project.
Okay, thank you. You should continue.
So we move on to part number 2 here, and here I'm going to describe SAXS studies where we're evaluating the pre-peak in SAXS. And probably as everyone's aware, small angle X-ray scattering, we have some type of a sample holder. We have the X-ray beam, and we're looking at the scattering of the X-rays by the solution to low angle, from which we get the momentum parameter here from the sine of 2 theta. These are experimental data for calcium and strontium chloride and also the bromide salts. This is what I would consider to be the wide angle scattering region. And these are the pre-peaks that we see in the small angle scattering region. So the observation of pre-peaks goes back as far as 1919. Paul Ehrenfest, looking at his photographic film, came up with these highly technical terms of the first strong diffraction peak that's observed in water, second strong diffraction peak. These are terms that have been used to this day, I guess, in describing these peaks with perhaps unknown origins. But it was understood early on in metals and metallic glasses that these peaks are often associated with hard sphere packing. And then after a few decades, it was found that the high concentration electrolyte solutions also show existence of these pre-peaks. I note that for calcium chloride at 30 millimolar, this is a physiologically relevant concentration, that this corresponds to, we see a pre-peak there at 0.2 reciprocal angstroms, which corresponds to a length of about 3 nanometers. Looking at this in a little bit different view here. So what we're probably more familiar with is the wide angle scattering region above about 2 or 3 reciprocal angstroms here. We just do Fourier transform of this region. It gives us the overall X-ray pair distribution function. This pre-peak first, excuse me, not the first peak in the pair distribution function is the strontium water distance at about 2.6 angstroms. The second peak is the chloride water distance at about 3.15. And then we have a third feature here, which is due to the solvent shared ion pair at 4.9 angstroms. What's less well known is the small angle region here, where we see the appearance of a pre-peak that moves to higher Q as a function of concentration. If we Fourier transform this, this is looking at the G of R. It's weighted by R cubed. And this is the partial due to the strontium partial. But what we see in the distribution of strontium in this solution, they are periodically ordered. And so this leads to the idea that there's domains on the order of 1 to 5 nanometers that have these periodic structures. We've done the measurements with a number of different electrolytes. This is one example for calcium chloride, where we look at concentrations from 60 millimolal up to 4 molar. And we measure the position of the pre-peak as a function of concentration. We have this plot here of the pre-peak position, Q naught, versus the concentration to the 1/3. This is similar to what's been observed in the literature by others. If you think of distributing the ions uniformly throughout the solution, the despacing between these ions will follow the concentration to the 1/3. So it's not particularly surprising here, but what's notable is that in dilute systems, getting down to 60 millimolal, we're getting to looking at length scales 4 to 5 nanometers. Another advance here is using molecular simulations to understand the source of these pre-peaks. So this is a study by our theory group where they use classical molecular dynamics to predict the small angle X-ray spectra for 3 molar strontium chloride. And we see these depending on whether you're using the chloride salt or the bromide salt, we get a different pre-peak structure. The beauty of MD is it allows us to deconvolute the contribution to this pre-peak from the various different partials in solution. For instance, in this upper right plot here, the dominant 2 contributions to the peak are the strontium partial and the strontium oxygen partial. So strontium is the divalent, it's strongly electrostatically repulsive. And around strontium, there's a very ordered layer of water. And so we get signals not only from the strontium correlation, but we get signals from the water around one strontium with the nearby strontium ions. So we have a strong signal from the cation. We see that the strontium chloride signal is anti-correlated, and then there's weaker contributions from the water, from the chloride partials. If you sum this set of partials here, you have the dashed line here, which is in good agreement with the, you have the solid line here, which is in good agreement with the experiment. Again, if you Fourier transform this S of Q, you can see the periodic oscillations in the strontium structure in the G of R. Another quite interesting system is the system of nickel chloride and zinc chloride in water. Here, interestingly, the ion-water distances between these two are almost identical. Their ionic radiuses are similar. Their Z values are about the same. So if we're looking at the contributions from the various partials of the metal, the chloride, and the water, they have about the same scattering values. The thing that's a bit surprising is that nickel chloride, at the saturation point, there's no contact ion pairs. And we know that from earlier XAFS studies. Whereas for zinc chloride, there's a complete contact ion pairing once you get above about 1 molar. And this comes from XAFS and V to C measurements. In fact, zinc chloride is sort of a model system for batteries. Zinc chloride is soluble in water up to about 20 molal. And this is really a decades-old problem in understanding electrolytes, is to try to deconvolute the contributions of this various species, the mono, the di, trichlorospecies in solution. And it's relevant to all of the transition metals, not only looking at the anions, various anions association, but the question is relevant to things like hydroxide formation. So it's a problem that has not been completely solved. There's a tremendous amount of information on XAFS, ZANES, and WACS for the zinc chloride systems. There's still large uncertainty in speciation. Recently, Jerry and his student DeWash did some X-ray emission spectroscopy, and this made significant progress in helping us reduce the number of variables here, but still not a complete solution. So it's interesting to see whether the pre-peak information can help us along with this resolving this question. These are the SAXS spectra for strontium, nickel, and zinc at low concentration. All of the systems are coincident that this is 50 millimolal. And in this case, the ions are fully dissociated. There's no interaction with between the cation and the anion. At 1 molal, we see that a nickel and zinc, the difference between nickel and zinc is that Q naught for zinc, it is a lower value, whereas at high concentration, the Q naught for zinc is at a higher value. So this is a little puzzling at first. At 1 molar, we know, of course, that in zinc, we have these multiple ion-paired species, and it's useful to think of, say, a zinc dichloride as being a neutral species when you're looking at the long range of electrostatic repulsion. So we're changing the distribution of the zinc chloride species, and it would be reasonable to believe that the effective Q naught would be at lower Q for zinc. The thing that was a little bit puzzling here is this shift to higher energy. We know, or to higher Q. So we know that from MD simulations predict the formation of these polymeric species where the zinc-zinc distances are shorter than they would be in the fully dissociated ions. So this would be one possible explanation why we see a shift to higher Q naught for zinc. So obviously this is a work in progress, but we're quite interested in how this SAXS region could tell us about the ordering. So we have very high quality measurements of the position of this peak, and we note that there's distortions in the symmetry of this that is correlated with the ordering of ions. So this is a case where we need to be informed from X-ray absorption studies about the short range structure in order to understand the long range structures. So I think this completes the second section here. And if we have banked any questions, I'd be happy to
We
answer
have banked
those.
just a few questions.
Okay.
Tim Pfister, you had a question?
Yeah, hi, John.
Hi, Tim. Yeah.
This is really beautiful work. We've done similar things with like acidic based solutions. And a trick we used to get to the very low concentration pre-peaks was to essentially subtract out the signal from uncoordinated waters. Is that something you've tried or?
That's what this is. So this
I
is
okay, it looked like it.
yeah, minus the pure water spectra. So if you're dilute, it's the perfect method. If you're using it at more concentrated solution, then it's not
Yeah.
as exact, of course. But yeah, that's our only hope of getting the ion specific signal out of the solution. Yeah.
Yeah, that makes sense. Do you
Yeah.
ever, at these higher concentrations, essentially run out of waters? Like, do you reach this kind of water and salt limit where, I don't know, things are either sterically hindered or you just don't have enough waters to coordinate everything?
Well, zinc is chloride is a special case, so
Right,
it's
right,
like
that one.
20 molal. There's I don't know, there's like two or three water, so it's not really a true aqueous environment there. But we've made measurements at high concentrations. We're first of all trying to just sort out the medium concentration range. But
Hmm.
yeah, yeah,
Cool, thanks.
All right, very good. Yang Ha, you had a question?
Yes, yeah, this has been interesting. So we actually look at a similar system. So my former colleague, Pat Frank, has done XAFS and also look at the Zane's regions to study the ion solvation. I myself also did some theory. And based on my experience, molecular dynamics does not do a good job on heavy ions such as bromides, where you need to consider the polarization, or some transition metal ions where the 3D orbitals come into play, and the covalency is something that you need to be considered. So my question is, since you have seen so many experimental data and you said you also have a theory team, do you also encounter something similar or during
so
when you compare your
kind of
experiments versus
yeah,
theory? Yeah.
here leading into the next part of the talk here. But yes, we've been looking at molecular dynamics for decades, and I am not very enthusiastic about classical intermolecular potentials. Leonard-Jones potentials are not appropriate for really describing the cation-anion interaction. We've had a lot of luck with DFT-based molecular dynamics, and it's a dramatic change in the fidelity of the simulation to do high-level DFT
DFT. Yeah.
simulations, both
Yeah.
using normal DFT and hybrid DFT functionals.
Great. Thank you.
Mhm. Yep.
Excellent. You should continue, John.
Okay. So the last section of the talk involves the locating the Kirkwood transition. So there's a theory that suggests that at moderate salt concentration, very roughly about 0.5 molal, that there's a transition from a Debye-Huckel-like distribution into these periodically ordered nanodomains. And we can look back in history now. Debye-Huckel is just a little over 100 years ago, and they made a solution of the Poisson equation for the electrostatic potential. They in order to make progress, they have some substantial approximations, but they came out with the screening length, this lambda d here, which is in common use today as a very easy concept to understanding the fields around ions. And the activity coefficients have been quite accurate for dilute electrolytes. And again, it was a triumph of theory 100 years ago. What's less well known is that John Kirkwood, this happy chap here, in about 13 years later after Debye-Huckel, came up with a more complete solution of these equations and found this remarkable result is that low concentrations, the G of R follows a damped exponential function in agreement with what you'd expect from Debye-Huckel, but at a certain concentration, you get the existence of damped oscillators. So we have the exponential damping term, and we have a cosine function that describes the oscillations in the G of R with distance. So this is shown in this conveniently in this plot here. So the red line is the inverse screening length from Debye-Huckel, and the black line is from the Kirkwood theory that shows at low concentration, very low concentrations, it agrees with Debye-Huckel, but at some intermediate concentrations, the inverse screening length increases, it goes through a cusp, and then the trend reverses. The other thing is the Q naught is the formation or the appearance of a pre-peak that moves to higher Q values as a function of concentration. So this has been a domain of theory, very active domain. Over the last 20 years, there's dozens of theory papers that confirm the expectation of this behavior. So we set out to try to find the Kirkwood transition. This alludes to the question from the audience previously about what methods are best for simulating correlations. And for contact ion pair, I'm a firm believer in DFT-based molecular dynamics. It's been a dramatic improvement in the fidelity of the simulations for predicting the contact ion pair species. Likewise, for solvent shared ion pairs, DFT is probably the method of choice. When we get to these long-range ordered systems, then we get to a very difficult choices, because the only methods that are available are classic molecular dynamics with very large simulation boxes with salt concentrations greater than 0.5 molal. Thankfully, the short-range structure is often not as important as it is for the contact ion pairs, so we can make progress with classical MD. But the thing that's really come to the rescue here as far as simulating these is using integral equation theory for these pair distribution functions, the equation known as the Ornstein-Zernike equation, and there's a variation of the closure known as hypernetted chain. This allows you to simulate very large length scales. You can look at infinitely dilute solutions, and it seems to be useful at concentrations below 0.5 molal. The input to HNC are the ion-ion PMFs. And thankfully, you can generate the short-range ion-ion PMFs from a DFT MD trajectory. So there's a lot of quality input and to HNC, and it seems to be working quite well at predicting these structures. We've made lots of measurements. The conventional technique is to prepare hundreds of solutions of different electrolytes at different concentrations, load them into capillaries, and run multiple series overnight or during the day. Another method that we've used to some advantage is to prepare two stepper motor driven syringe pumps. We have electrolyte in one, water in the other. We can set up a continuous ramp, and then we can continuously measure the scattering from these solutions. And to give you then the quick anatomy of this pre-peak, one of our favorite solutions is erbium chloride, and erbium is a very high Z cation, and so we get very strong scattering from the erbium. In fact, if you look at these solutions in the 1 to 5 molal region, this peak is so intense that it dominates the entire small angle and wide angle scattering region. As we increase the concentration, we see the appearance of the pre-peak, and it moves the position of this peak, Q naught, moves to higher Q as we increase concentration. So Q naught is correlated with the ion-ion periodicities. The width of this peak is equivalent to the inverse screening length. This is how we informed from HNC theory. This is what would be predicted for the Kirkwood transition. Notably, the dozens of theory papers mainly deal with 1,1 electrolytes in solution, but for sodium chloride, for example, the Kirkwood transition is predicted at about 1 molal is where we expect to see the onset of the pre-peak. For divalent cations and for trivalent cations, the Kirkwood transition moves to lower and lower concentrations. So for strontium chloride, we expect the Kirkwood transition to be at about 0.2 molal. And as just a little side picture here, this is output from an MD trajectory. Again, this is classical MD using empirical potentials. And this is a box. It has 3 molal erbium chloride. There's 10,000 waters, and it's a 20 nanosecond trajectory. And in this frame, what we're looking at is these little purple spheres represent the path of a single ion over the trajectory of about 5 nanoseconds. So we can see these little grape clusters here that show the motion of a single ion in time. And in this circle I've drawn here, you can see roughly a 3 by 3 box here. And if you take away the green spheres, which are the chlorides, you can look down into the solutions and see that below the top surface, we have other erbiums that are ordered directly below. So approximately, this cell is a 3 by 3 by 3 cell. This other circled domain here I have on the left is perhaps, instead of a face-on view, it's the cell tilted at 45 degrees. So well, I have to be very careful in defining what these are and what they are not. So to be very clear, these are not crystallites in solution. So they're fluxional, they're disordered, they're quasi-periodic in nature. They have d-spacing, in this case, of about 1 nanometer. And you can also talk about things like spatial coherence length, which is about 2.5 nanometer, which is derived again from the full width of half max of the pre-peak. We've done measurements of strontium and erbium chloride. This is the experimental data showing the transit of the pre-peak. The pre-peak is a function of concentration for both strontium and chloride. And this is a plot showing the how the A naught changes. So as you increase concentration, we get an increase in the inverse screening length, which is in accord with what you would expect from a Debye-Huckel response. But at some finite concentration, this turns over and we see a decrease in the screening length, both for strontium and for erbium. Likewise, the pre-peak, as we increase concentration, we see sort of a sudden step appearance of this pre-peak, and then as we increase concentration, Q naught values increase. So if we compare this to the HNC theory, to the right here, the blue line is the prediction of Q naught from HNC. The pink crosses are the experimental measured values. So we have good agreement with sort of a plateau region, a drop in Q naught down close to 0. Likewise, for A naught, we see an increase, a peak, and a decrease. The experiment in this case is not quite as closely matched to theory, but it still shows the existence of a maximum in the experimental data, and a very similar observation for erbium chloride. So we think this is good evidence of a Kirkwood transition, at least for these divalent and monovalent, the divalent and trivalent salt. So finally, this is a plot. Sometimes you have scientific data that rises to the level of a piece of art. And this is a prediction from, you can see, signed by the artist here, Greg Schenter. And this is the prediction of the Kirkwood transition for sodium chloride. And what we're looking at here is the R-weighted absolute value of the G of R. So this H function is just the sodium pair distribution function -1, and we're looking at the absolute value of this cosine function here. So as the cosine value passes through 0, we get these cusps here. And what this shows very elegantly in this type of presentation is that for sodium chloride at low concentration at 10 millimolal, we have an exponential decay in the G of R. And when we get to about 0.8 molal sodium chloride, all of a sudden we see the onset of these oscillations. As you go to higher concentrations, we get an increase in the periodicity of the structures. So this would conclude my talk, and to note that if you want to look at the chemical response of any of these aqueous systems, you need to know not only the short range structure, but the long range structure. And in many electrolyte systems, values very roughly around 0.5 molal. Electrolytes exist in these ordered nanodomains. We've used a combination of experimental and theoretical methods to pinpoint the location of the Kirkwood transition. And then finally, I'd like to point out that in order to make progress in the long range, we need X-ray absorption spectroscopy to look at the short range structures. So I would think that there's merit to X-ray scattering and suggest that perhaps we need to move ahead with a new series, the X-ray absorption and scattering spectroscopy journal club. So with that, I would be happy to answer any questions.
That was wonderful, John, and I look forward to seeing your announcements for the new journal club. So we'll start with a question from Fairvila.
Hey, I have a couple now. First of all, when you're doing the classical MDs, are you using a polarizable water potential, or because I mean, those are very big cells. That's number one.
Okay, so I think it's my guess. I don't know. It's probably SPCE water, and
Okay.
the ion-ion pair potentials are adjusted at long range to conform with the Coulombic repulsion or attraction at very long scales, and the cation-anion interactions are typically come from the mixing rules, but they're sometimes adjusted to what the experimental values are.
Okay. So the other
Yeah,
question was, sorry.
go ahead.
Okay, no, the second one was, I mean, you have this beautiful fluxional quasi domains in those pictures, and maybe I know you want to call them fluxional, but I mean they almost look crystalline. So is there any way you can predict the macrostructure of those things by looking at like effective charges and like long range Coulomb interactions and these things to just essentially predict something like a crystalline structure in this?
In this paper that I provided, we used order parameters in the MD trajectory to try to identify whether it's, for instance, a cubic phase or cubic-like, I guess. So evaluation of the order parameters suggested it trends towards certain symmetries.
Okay. Okay. So you didn't go any deeper than that, than just order parameters.
I think that, again, I can't speak completely to the theory side, but I
Okay.
believe that that's one of the things that we've done.
Okay. Thank you.
Mm-hmm.
Okay, next question from Insu Lee.
Hi, John.
Hi,
Long time no
Insu.
see.
Yeah.
Yeah, nice work. But I think those metallic solutions, usually when we mix that at our beam line trips, we had to adjust the pH, right? But I'm
Yes.
not sure if those solutions are so like dilute that you had to do pH control? And
Oh,
yes.
I didn't mention, but for all the transition metals, and especially for trivalent cations, we always adjust the pH to 1. So they're
Okay.
low pH, and we use the HCl, but the added chloride concentration is not really significant to the total. But for sure, in order to prevent hydrolysis, they must be at low pH.
Okay, so hydronium ions would not, like, I don't know, different pH values would not distort the structure. Would it have an effect,
Well, the
or is it negligible?
hydronium concentration is still quite low. So at pH 1, I guess, relative to the total salt concentration, it's 1. And yeah, I think that overall the hydronium concentration is a small percentage of the total cation concentration.
Mhm.
Yeah.
Okay, Matt Newville is going to manage the last couple questions. I have to go to a student's general exam. It's awesome talk, John. I got to run, but Matt's got you.
Okay.
Great. Thanks. So Yang, hi, you had another question? So,
Yeah, it's a pretty general question. It's like when I do this theory, people always ask me, like, can you use your theory or experiments to predict the solubility of different ions? Does that work in your system?
The saturation, concentration, nucleation, these are all very active and difficult questions. So um, I think we're not the focus of our research is not solubility, but certainly that is a critical question in electrolyte solutions. This is also sort of a different concept. When we think about saturation, concentration, solubility, we tend to think of uniformly distributed ions, whereas in fact, for a lot of these electrolyte solutions, at saturations, we have these domains. So this is something to consider when you think about solubility and nucleation.
Thanks.
okay, I think I want to go back. KY Zhang, did you have a question about lifetimes? And
Yes, I did. Thank
it
you.
sounds fascinating, so I wanted to.
Yeah, so my question is, when you see an ion pair on XAFS or on X-ray scattering, do you know whether this is formed by like random motion that brought together cation and anion and form a transient ion pair, or there's stronger interaction, maybe Coulombic interaction that forms a more stable ion pair that has a longer lifetime? Can you tell that?
Well, we're not, of course, with XAFS or X-ray scattering, these methods, we can't measure directly the lifetimes. We know that DFT-based MD is very high quality structural, and the lifetimes come automatically out of that. But it depends on the degree of ion pairing, of course. But I think water. Just looking at water as a ligand on an ion, the lifetimes can vary from nanoseconds up to hours. In the case of small trivalent cations, the water lifetimes in the first shell can be many hours. So I would kind of expect that we would have a similar response for some of these ion pair species.
Okay, thank you.
Great. Tao Li, did you have a question about?
It's John. Very nice talk. I have done a lot for this using SAXS look at the lithium ion electrolyte, but we mostly focus on the lithium, not like you high Z material. So we cannot see the the zinc and other things. I have a question for your system. We saw a lot of pre-peak. We even sometimes more than 2 pre-peak. So for you see the clustering for your cations. And how about the water, the solvent? Do you also see any clustering or not?
So let's see if I can find the this is sort of a breakdown of the contributions of the various partials, of course.
Yeah.
And the two dominant ones, of course, this strontium is the highest Z species atom in the solution. So in the X-ray structure factor, it's dominates the signal. But again, the strontium correlation and the strontium oxygen, the water correlations are the major contributors here. And we see minor contributions from water chloride. So that gives you a sense for, you were saying that you have lithium. What is the count in your system? Lithium, what is the counter ion?
PFSI.
Oh, okay. That's a polyatomic anion with fluoride and
Right.
sulfur, right?
Yeah.
Right. So that in that case, your signal is dominated by scattering from the anion. So
Right.
there's very little scattering from lithium or even from the water. So yeah, again, it's just this simple set of contributions here. In your case, you'll be looking primarily at the anion contributions when you're making this measurement with X-rays.
Yeah, correct. And we also do the neutron scattering to look at the solvent.
Yeah, yeah, neutron is good for lithium, in fact. So I guess there's a good scattering cross section for lithium, so you can hope to pull out that partial.
Yeah, correct. Yeah. Yeah, thank you, John. Yeah.
Yeah.
Oh, okay, we're close to the hour, but I think there's one more question from S. Catherine. I don't recognize you. Do you want to ask your question?
There was no question. I was just answering some of the people's comment. Some of the people in the chat were wondering about force fields. So I
Great. Okay.
was just trying to back John up and provide some details there.
Well, thanks. I think we all appreciate that. So thanks. So if there's no more questions, let's thank John for a wonderful talk. And yeah, so thanks. And I'll stop the recording. And